Hi everyone, this is Jacob Jutenik, and today I'll be presenting some information and research on Parkinson's disease for our Frontiers in Neuroanatomy presentation assignment. Let's get started. First things first, what is Parkinson's disease? So Parkinson's disease, or otherwise known as simply PD, is a progressive degenerative brain disorder of the central nervous system. This disease primarily worsens motor functions and thinking while causing severe dementia and depression anxiety over time due to a lack of dopamine neurotransmitter in the brain. You can see in figure 1 to the right there a nice comparison of brain activity between a healthy normal brain and a brain with PD symptoms. This shows specifically that it seems to happen in the neurons in the middle of the brain or the center of the brain, but we'll get into that in a second. First things first, we need to realize that the cause of PD here is really unknown and can't seem to be pinned down to just one thing. However, it appears to arise due to some combination of genetic and environmental factors. We also need to realize that there's no cure for PD, so instead, the treatments aim to diminish symptoms and improve the quality of life overall. So, what really happens during Parkinson's and what symptoms seem to appear within these patients? Well, PD destroys the dopamine producing neurons in the substantia nigra, which is the small blue portion shown in figure 2 there, and it's found tucked in the midbrain. That portion is a basal ganglia structure that is responsible for dopamine production, among other functions involving reward and coordination. So when this disease destroys cells in this region, it ultimately diminishes dopamine production and causes issues with balance and movement. So scientists believe that an accumulation of abnormal alpha-synuclein protein in the brain called Lewy bodies may play a part in killing the neurons. So when these Lewy bodies are found in the substantia nigra neurons of PD patients, such as in figure 3 there, um, they believe the bodies slowly kill off other cells over time and spread to other cells in the process. And this eventually leads to the development of symptoms such as tremors, problems with balance, hallucinations, stiffness in the limbs, advanced dementia, depression, anxiety, etc. Now, how can we treat PD? Because as I mentioned previously, there is no cure for PD, and the treatments just simply work to limit symptoms without stopping the disease in its progression overall. Uh, so for treatment, they try to use a combination of medications, and some medications, such as L-DOPA or levodopa, work to increase the level of dopamine in the brain by acting as a precursor for dopamine, in which the neurons can convert into dopamine. Other ones, such as MAOB inhibitors, slow the enzyme responsible for dopamine breakdown, meaning that they'll keep the dopamine that is already in the system. And finally, anticholinergic drugs help to control the tremors and the muscle rigidity. And, usually overall, these treatments have some combination of these medications based on what their symptoms are. So that's kind of how we can treat this disease. Now, before we get into the research, I thought it would be nice to share some fast facts on Parkinson's disease. And first, there are 5 stages of PD, with stage 1 being the first and the least critical stage, and stage 5 being the final and worst stage. The early stages seem to not show many symptoms, mainly because when approximately 80% of the dopamine producing neurons in your brain are damaged, the symptoms of PD begin to appear. And this is why typically symptoms aren't bad or even noticeable until later in the progression, such as around stage 4 or 5. As common as this is, up to 1.5 million people in the United States are believed to currently suffer with PD, with approximately 100,000 new cases of PD diagnosed each year. That prevalence can be seen in figure 4 there, and it shows that a lot of these cases are found on the East Coast and actually kind of the Northeast coast and the northeast side of America, which I kind of thought was interesting. I also found it interesting that the average age of onset, so the average age that patients are diagnosed with Parkinson's, is 62, meaning that the disease seems to take effect much later in life for some reason. Anyways, on to the PD research article that I picked. So, the one I decided to use for this PowerPoint was published in December of 2017 by 4 researchers from the Ciudad Real Medical School of the University of Castilla-La Mancha in Spain. I put their logo on the slide just because I kind of thought their university was cool. Anyways, this team focused their efforts on the role of the anterior olfactory nucleus, or the AON as they abbreviate it throughout this article, and the olfactory bulb as indicators of the initial stages of PD. Their goal was to analyze whether visible alpha-synuclein Lewy bodies of the olfactory system were involved in the early vulnerability of interneurons associated with Parkinson's disease. So, in their introduction, they noted that prior research into the subject revealed that the anterior olfactory nucleus, or AON, as I'll abbreviate it from now on, and the olfactory bulb were involved in stage 1 Parkinson's symptoms, such as the olfactory degeneration, hyposmia, which is a lack of smell, REM sleep disorders, constipation, dysautonomia, which is when the autonomic nervous system doesn't work correctly, um, etc. So figure 5 here shows the AON and the olfactory bulb in comparison to everything else, and so it's definitely an anterior structure in the brain. And the AON also has 3 different portions that are studied here that you'll need to know throughout this research, which are the retrobulbar, abbreviated the AON-RB, the cortical anterior, abbreviated the AON-CA, and the cortical posterior, which is the AON-CP. The olfactory system overall, and specifically the AON in each of those 3 components, had previously been determined to be an early site for Lewy body development in the brain. And using this information, the team hypothesized that the AON is an early and preferential region of initial alpha-synucleinopathy in PD due to its peripheral location and multiple short and long distance neural connections. So, how did they pull this research off? Well, brain tissue specimens were taken from 4 neuropathologically diagnosed cases of PD, each of which were in stages 4 and 5, and 4 age-matched controls alongside of these. All of their information, all of the 8 patients that were used in this study, um, um, shouldn't say used, observed in this study, if you will, their information is shown here in Table 1. And we can see here that all but one of the participants were male, and most were entering an older stage of life, such as in their 50s and 60s, 70s. There's even an 80-year-old here. And those with Parkinson's disease were in diagnosed stages 4 or 5, like I mentioned. So after they collected these specimens from these 8 patients, they used a freezing sliding microtome to create 10 series of 50 micrometer sections for each specimen. The first specimen was Nissl stained. The second was used for immunohistochemistry against the alpha-synuclein. The third and fourth were used for immunofluorescence analysis, and the rest were kept in case they needed repeats or additional study on the material. Furthermore, the AONRB, CA, and CP divisions that I mentioned earlier were then identified in parallel Nissl stained sections as seen here in Figure 6. The AONRB and CA were shown to have olfactory degeneration in this figure, specifically in the black box portion in the middle there. They kind of pointed that out, that's where it was. And these were then analyzed using ZEN and ImageJ software from the NIH. Co-localization as a percentage of total cells was then obtained by comparing the total number of cells expressing a neural marker to the number of cells that also co-expressed alpha-synuclein. So, this is how they did it, and they found this stuff. So, the team found higher expression of parvalbumin in the neural cell bodies of all of the AON divisions of PD cases compared with controls. And you can see this in the top part of this figure, figure 7 there, where the PD column for parvalbumin is significantly higher than control for all of them. Their PD value for this portion there, which is labeled PV, was 0.00001 or less for each of them. So, very significant. They also found higher expression for all calcium-binding proteins except somatostatin in the cell bodies and neurites of neurons in the AONRB of these PD patients, and specifically the calbindin, which is the CB there, had a p-value of 0.0178. Calretinin there, which is labeled CR, had a p-value of 0.0366, and paravalbumin, again PV, had 0.0001 this time. These are all shown in the bottom of the figure here, where PD again surpasses C for all of these columns. Finally here, there was also a significantly higher expression of parvalbumin in the neurons and of neurons in the AONCA of PD cases here as well, with that p-value being 0.0031. So, the additional results then found the neurons expressing the calcium-binding proteins had the highest percentages of alpha-synuclein co-localization, as shown through the light portions in each AO1 division, seen here in Figure 8. Somatostatin and alpha-synuclein positive cells were rare, although there were higher percentages in the AONCA, as you can see here in the C and D part of this figure. And again, these are the boxed portions here in this figure, kind of the zoomed in parts you can see there. So anyways, the co-localization of alpha-synuclein in the AONRB, CA, and CP with calretinin, calbindin, parvalbumin, and somatostatin is shown in the figure, and ultimately finds that alpha-synuclein, and well, it finds alpha-synuclein in each of these divisions here, which shows symptoms of Parkinson's disease. So, overall, the team found that increased expression of calcium binding, particularly parvalbumin, correlated with alpha-synuclein co-localization in the different divisions of the AON. Data from this experiment supports the idea for differential regional alpha-synuclein involvement, and therefore that the divisions of the AON need to be independently studied. The results also suggest a potential differential vulnerability that may be due to alpha-synucleinopathy among interneurons populations in different divisions of the human AON. It's unclear to what extent cells expressing calcium-binding proteins can buffer the pathological effects of alpha-synuclein and somatostatin-positive neurons, which the team hopes will eventually be covered in future studies. So, here are my references, as well as a huge thank you for listening to my virtual presentation here. And if you have any questions, feel free to reach out. Stay safe out there, everybody, and thanks again.